Today we're going to finish off that last lecture, and I'm going to introduce you to the website where we're going to do a lot of the lab material. And just a reminder that here's the syllabus, which contains information about the labs and also contains information about the assignment. We're going to have more information coming up about the assignment and the labs soon. The labs are going to be completed each week in class, and you're going to hand in that week a particular assignment through e-class, which will usually be the uploading of a couple images, like charts that you'll make from Excel. And the instructions will also be in videos or in written documents, and so if you miss the lab itself, it would be possible to catch up, although you wouldn't have access to the EEG devices anymore. The assignment, as we said, is going to be based off some data we collect in the lab, and the assignment's going to be done in groups of 1 to n people, as many as you want to have in your group. The labs are going to be done in pairs as well, and we'll probably assign those pairs to keep everything flowing quickly. So today I wanted to just talk about the material for the lectures, and I wanted to quickly go over the website eegedu.com that we're going to be using for the lab, and I'll direct you guys to start following along with the first module, which we're not going to cover in the lab, just an introduction to the device and what we're going to do. So here I've created 2 folders now, the lecture notes folder, which has in it all of the Word documents of the material I want to cover each week, and then I also have here the lecture slides, which is a new folder now. It's going to be a folder. For now it's just a single document, and so this is going to be now Google Docs instead of PowerPoint slides, and you'll see that this will make it easier to present. So I'll just jump right in. So we'll view this as a slideshow, and so a lot of this will be just a recap from what I covered last class, so you can skip forward, but I just wanted to cover some of these more difficult things one more time. So recall that the brain uses glucose and oxygen from the blood when it is active, so it needs to recruit this blood to specific areas of the brain that were active, and we can use this blood change as one of the signals in our neuroimaging devices. Remember also that neurons become depolarized in big groups together, and that these depolarizations can create oscillations in electrical activity that can be read nearby from groups of electrodes. And we've seen that the spiking of individual neurons, that is when they fire action potentials, can be linked to the ongoing oscillations in their electrical activity, because their resting membrane potentials oscillate as well, and the threshold for firing will only be reached during certain cycles of that oscillation. So for example, even in humans, we were able to show about a decade ago that depending on which phase of an ongoing alpha wave someone is presented with a circle, a very faint flash of light, they're more or less likely to see that. We'll talk again about that later today. We've talked about other ways we can electrically record from human brains by implanting electrodes on the surface of the skin or deep within the skin. And then one last thing I wanted to recap again, because it's difficult, is this idea of MRI physics. So magnetic resonance imaging measures the energy given off by big groups of protons that are in our heads and bodies when we align them in a magnetic field, and then add a little bit of energy to knock them into a different alignment, which then gets given off as they bounce back to the original alignment. And depending on the tissue qualities, the signals, these kind of yellow electricity looking things, can have different properties. And those different properties, maybe they're faster to flip back in different parts of tissue. So here, tissue with water, like our cerebral spinal fluid, is shown as darker, and the gray and white matter is shown as lighter because of different properties of these electromagnetic signals given off when they're all aligned in one of these magnetic fields, radio frequency gets added to flip them, and then they give off that energy as they flip back. We talked about last class how this technology can also be used to estimate the trajectory of water through tissue, and in turn can be used to estimate white matter tracks through the human and animal brains non-invasively. We call this diffusion tensor imaging because each voxel of the image is attributed one of these tensor shapes depending on how it's like this, with a high directionality to the way that water flows through them, much like set top down. Again, another reason signals can go down into these lower visual areas and modulate spatial and have other properties. And by light, for example, to observe what it is that makes certain parts of the visual cortex of the monkey more or less active, in that there's more or less blood flow at certain times. Picture of exposed cortex that has light shining on it, and because the light gets differentially absorbed by areas with images like this comparing two conditions, or images like this which are composites of maybe 10 images, showing in this case ocular dominance columns, and in this case columns, that is that specific that seem to be arranged in these pinwheel shapes. Is in 275 and 375, the dorsal pathway, which allows ventral pathway, which identify two things can happen to the light. One is that the blood in the area can differentially absorb different colors of light, so you can recreate maps based off which light sources and which light detect which areas were more versus less active. Near infrared light comes back to the detector blood in this way because oxygenates neurons in the left visual. Optical signal is associated in time with electrical activity. As changes in depolarization of neurons' membranes occur, there